Hi, I'm Joe Lewcock, and I'm head of biology discovery at Denali.
I'm Valina Dawson. I'm a director of neurodegeneration and repair in the Institute for Cell Engineering at Johns Hopkins.
And we're here to talk about the Keystone Meeting, neurodegenerative disease, new insights, and therapeutic opportunities.
Which has really been a terrific meeting. I think we accomplished all of our goals that we set out originally.
Yeah, and it's been a long time planning the meeting. I thought it was a really fun concept to really bring together a lot of different disease areas and different types of biology into one big group.
Exactly, and to have this also in conjunction with the glial component, because unbeknownst to us, that glial component became a really big deal this year in neurodegeneration.
Yeah, definitely. That biology was a big part of the meeting. And, you know, coming from the industry perspective, it was also really great to see a really good amount of industry engagement and a really good mix between industry scientists and academic scientists. And I felt like there was a lot of crosstalk there, which was encouraging to see.
Well, absolutely, and a lot of the scientists now are moving to try and understand those key therapeutic targets, and they also want to develop molecules that will go into clinical treatment. It was good to have industry here to help talk to them and provide insight and advice, on how to do this. But also, particularly your company and Ionis are doing amazing things with the ISOs and really detailed mechanistic targeting was really pretty exciting to see.
Yeah, and I think it's really great to see, that bridge with academics where it's really that mechanistic work that's done in a lot of the high quality academic labs that have been here that have started to enable these new therapeutic strategies. And so, really bringing that together has been quite exciting. So for you, when we started thinking about this meeting, I mean, what were the things that you really wanted to make sure that we talked about? What did you feel like the most scientifically important topics?
Well, initially, it was this idea that many of these neurodegenerative diseases have a common feature of misfolded proteins, and that this understanding now that these are inherently disordered proteins, and that they seed, template, and transmit. And this is not unique to one neurologic disease, but it seems to cross over disease entities. And I wasn't expecting it, but really that was addressed on a lot of the talks. People who were coming from a variety of very well-known diseases, from Alzheimer's to Parkinson's, to less known diseases or rarer diseases like ALS, all had this similar theme of these disordered proteins. And then how do we understand and build models knowing this now? Yeah,
I completely agree. And I felt like, although there was some discussion around A-beta and A-beta related therapies, which, have had mixed success over the years, it was really exciting to see a little bit more on proteins like tau, where I think it's becoming much more well accepted that these things do spread from cell to cell. They spread throughout the CNS for both tau in the context of Alzheimer's disease as well as alpha-synuclein in the context of Parkinson's. And really going from a few years ago having that be something that I think was hotly debated whether that was a relevant mechanism to something where not only are academic labs really pursuing the molecular mechanisms that underlie that spread, but there was a number of different companies coming with, talking about therapeutic opportunities and therapeutic therapeutics, let's say, coming with different therapeutics, targeting this spread, and one presentation from Karina Fogg at Lundbeck, where she really highlighted, the massive amount of work that they were doing, looking to find the optimal antibody that would block synuclein spread through the CNS. And, I felt like that was a great complement to some of the basic science work, including your own, showing exciting models of, how we might better understand how that spread occurs in disease.
And then the group out of Pennsylvania was really starting to highlight the fact that most of these diseases have comorbidities. So you don't just have A-beta, and you don't just have tau or synuclein pathologies. You have a mix, and they're starting to build models with these mixes. And then today with Karen Ash, she was in there showing mixing genetic models for tau and A-beta and really showing that these molecules can interact with each other in a pathologic way. And that really has not been something we've seen a lot of before. It had been, one molecule and one disease, and they were separate from everything else. And now there's this idea that they're probably a sliding scale.
Yeah, I completely agree. That was a great talk from her this morning, and really taking it through her own evolution of how she saw the field change during the course of her career, and then having some of the young scientists from Virginia Lee and John Trojanowski's group really highlight some of the new models that they've been doing to try to understand the interplay between A-beta pathology and TDP-43 pathology, or A-beta pathology and tau pathology. I thought that was a very exciting.
Right, and then they pulled in Huntington's disease, which everyone thought was completely separate, and showed that there were common molecules mediating synaptic toxicity and dysfunction. Yeah. So that was pretty amazing.
Yeah, and it was great, and I think, for me, being at the meeting, it was really a nice for us, a feel good for the approach of how we set up the meeting, to say, let's bring all of these different disease areas together, and have people talk about their science, because even though, there do seem to be these shared mechanisms, some of these fields don't always talk to each other about the exciting work that they're doing.
And from this came a lot of collaborations, because you read your own literature, and you don't, there's just so much literature out there. And one common thing I heard from attendees is that they were setting up new collaborations, and they were very excited about having come to this meeting, that they not only did they get some new knowledge, but they're now getting new tools. It's exciting. And I always say for myself, I definitely am coming out of here with a to-do list of people to follow up with, which is great to see. What did you think about the some of the talks surrounding the role of microglia in disease? Now, that was an interesting component to this, because in the past, neuroscientists have really ignored the glial component, and we really thought that microglia just were there to clean up the mess. They got activated because there was some kind of debris, and that glia were just supportive, but they weren't really active. And what really came out of this meeting is that microglia and glia are really early participants in the disease process. They may even be initiating the disease. And that was a surprise, but it was a common theme, that these big neuroscience labs are now incorporating a glial component to their labs, and that they were then running over to the other meeting and vice versa to get advice and get some new knowledge. But that's clearly going to be the way that we're going to move forward, is that we're going to have to understand all of the compartments, both the glial and the neuronal compartments, to really understand the full pathology of these diseases.
Yeah, and I agree, and that there's definitely been a paradigm shift, and likely part of that has been the recent genetics linking microglia, to Alzheimer's disease in particular, but also schizophrenia and some other diseases, and really exciting to see, I think, a growing attention on this cell type, but then also some interesting new technologies that are enabling a much deeper understanding of microglial biology. In particular, I can think of Eric Huang's talk when he was looking at the progranulin knockout animals, and he had done, just a massive amount of work looking at single-cell RNA-seq from multiple different time points, and really getting a very deep understanding of how microglia were impacted in disease. And one of the things that was interesting about that talk to me is that although it does seem clear that they're involved in disease, they're likely not involved in the same way in every disease. It's not like a one-size-fits-all type of thing. And so I think in the coming years, it'll be really exciting to see continued advancement in our understanding of that cell type.
And I know that they're starting to work on trying to characterize and differentiate each type of activated microglia. So it's gone beyond microglia are activated to we have, the M1 activated microglia, the DAM, the lysosome. But even within that small group, there are going to be further differentiations and characterizations, and that field is going to move in that direction, which will be helpful in understanding disease, and then also understanding where you can put the therapeutics in.
Yeah,
You know?
I agree. And from a therapeutic perspective, I think, we're just beginning to understand how cell biology is misregulated in microglia. And, really hopefully in the next few years, that will lead to exciting therapeutic opportunities that are really based on, not just that these cells are activated and you want to turn them off, that it's not that simple, and that there are going to be more specific strategies that we're going to want to do to target this cell type in disease.
Well, and particularly with Beth Stevens' talk, where the microglia facilitate synaptic remodeling, but then get a little unhappy and facilitate the disease. And it was surprising that she was able to take an antibody to just one complement and really restore neurobehavioral function in animals.
Yeah, that was very exciting. And I think that, those types of therapeutic approaches have a lot of promise. I was also really excited about hearing from Frank Rigo about the work that Ionis Pharmaceuticals is doing around antisense oligonucleotides, or ASOs. I thought that, I was just so impressed by how the types of things they're able to do to modulate diseases. And once the genetics of a disease have been identified, how they can really take a very targeted strategy towards it. And it was exciting to see him do it, but it was also exciting to see that the use of these ASOs had also spread to many academic groups who were using them both as research tools, but also to validate them as potential therapeutics in the future.
Absolutely. And there are some very clever ways that they were using the ASOs to regulate splicing and expression that were somewhat surprising, but absolutely essential for those diseases, because there's no way you could drug those diseases in any other way.
Yeah.
So it was really like taking a fine scalpel to the disease process and then completely eliminating it. It's exciting.
Yeah, I mean, definitely. And to see the first effects now translate to the clinic with Spinraza and also the antisense against SOD1, I think there's real promise to say that this is not just something that you can do in the lab, but that is potentially clinically translatable.
Oh, yeah. And it's made a tremendous difference in the lives of those children, too. So
yeah.
That's nice.
And then I was also really impressed by some of the new research tools that are being developed to really enable, I think, new discoveries that might not have been possible just a few years ago. And one example being a great talk that Rusty Gage gave, and really talking about being able to differentiate directly to neurons from fibroblasts from Alzheimer's patients, and doing that with multiple patients and being able to really better understand some of the phenotypes that these sporadic patients present.
And making that distinction between reprogrammed iPSCs, which are a young cell that have, and they have a lot of potential and opportunity for doing mechanistic biochemical assays in them because they're readily renewable and you could generate lots of them. The direct reprogramming, you generate less cells, fewer cells, which is a problem. But they maintain their aging phenotype. And the fact that he was able to distinguish and differentiate that aging from Alzheimer's, that Alzheimer's not due to aging per se. So if you get to 102, you're not guaranteed to get Alzheimer's, that it's a disease separate from aging.
Yeah.
And I thought that was really important and very key.
Yeah, definitely. And I think it is, as you said, very much a lesson learned that going to iPSCs, you're only getting part of the story, most likely, with a lot of these diseases being age dependent, that you may not reflect key aspects of the biology. So I felt like that was a really important lesson that I learned out of that presentation.
And he also brought up the future need for that area for tool development or technology development is to be able to take those inducible cells and make organoids out of them, so that you can then have a three-dimensional tool to work with and to be able to add in exogenous microglia and start to understand the aging disease process in a three-dimensional space.
Yeah, agreed. And I think many labs seem to be moving in that direction. Ranging from organoids to even grafting human cells into a mouse brain, which Matthew Lurton-Jones has talked about, and I think it is super important. And having worked on microglia myself, and knowing that microglia in a dish compared to microglia in a brain are a very different thing, right? And so to be able to create environments where it's more reflective of an in vivo situation is going to be super important. And
then it offers the opportunity to use that whole array of CRISPR technology to really molecularly dissect what the microglia are doing and what are the important pathways in them.
Yeah, I definitely agree, and I felt like Martin Kapman's talk from UCSF was a great example of that, where he's able to start mixing some of these iPSC technologies, both iPSC-derived neurons and other cell types, and then doing whole genome CRISPR screening, which seems like a Herculean task, and I'm sure it is, but it's an amazing way to go from to get a better understanding of the pathways that are relevant in disease. And I feel like that's going to be super important in the field of neurodegeneration, where often you have, genetics that help contribute to disease risk, but the mechanism by which they do it is not entirely clear. So to be able to, take these types of technologies and really better understand those pathways, I think is going to be has big potential for the future.
And I think these transcriptomics are opening that up. They're providing some new insight and some surprises. And then they're also providing commonalities. And so people are starting to think about what are the main key pathways and how do you intervene. And surprisingly, they're not that different between the diseases. They may get a different start, but then they start to converge. And that offers a lot of opportunity for intervening. So.
Yeah, I agree from a therapeutic perspective. I think that's really exciting. And, to really, if one can identify those shared pathways that are relevant across diseases, those would make very attractive therapeutic targets, for multiple different types of modalities, ASOs, or even more conventional small molecules, or perhaps gene therapy. So, I'm hoping that that's going to open up a lot of doors in the coming years for potential therapeutic approaches.
And then what did you think about these topics looking at development versus aging and these day jobs changing from development to just normal day jobs and then into disease? And
I thought there was a number of exciting talks about that, and it's an interesting concept. That's sort of why reinvent the wheel, right? That if there are molecular mechanisms in place, that those can then get co-opted in the context of disease. And I think it makes sense, but it's something I haven't heard that much about before this meeting.
Yeah, and I thought it was good to see that some of the investigators are starting to say, we know like tau is toxic, but what does it normally do?
Yes.
And really kind of starting to understand and learn new things about that protein that, we've had decades with.
Yeah, definitely. And I think there were some great talks about tau, synuclein, and TDP-43 in that regard. Looking at some of the, as TDP-43 is an RNA binding protein, and people have focused on the pathology more than the normal function. But, now getting that understanding, and there's a couple of talks that touched on this, that understanding that when you do have misregulated TDP-43 in ALS, that does result in downstream effects and altered splicing. And the story on Stathman-2 was particularly interesting, where it really showed pretty major effects on gene expression.
Yeah.
So, Valina, we talked about, I think some of the exciting opportunities for the future. What do you think some of the challenges that field is going to face in the future are?
Well, I think one of the exciting talks dealt with sex as a variable, and there were some really surprising outcomes by gender. So particularly Lee Gann's work with microglia and the TREM knockouts was totally surprising that the female microglia behaved very differently from male microglia. I mean, who would have thought?
Yeah, and I agree, and it's something that I think currently investigators don't build really into their in vivo studies or in vitro studies. It seems complicated enough to deal with all the other variables, but now to know that gender is going to be something that may make a difference, I think it's something we definitely want to take into account moving forward.
Absolutely. What did you think were some of the challenges?
Well, I mean, I felt that one of the really impressive things about the conference was the massive amount of data that people were generating through single-cell RNA-seq or nuke-seq, and really, getting much deeper data sets than we're used to. And I think one of the challenges moving forward is going to be how to deal with all of that data, how to store it, and how to be able to compare data across labs. I think everyone does these types of experiments a little bit differently. And so understanding with these huge data sets what represents variability in the biology versus variability in technical aspects of how they've done things will be really important.
And I did hear from some of the speakers as well as the attendees that we also need to be able to present that data in a meaningful way.
Yes, yes, and I think that can be a challenge because it's a huge amount of information to fit into sometimes a short talk, right? From an excited young investigator, yeah.
Yeah. The other that I heard, because people are looking at these misfolded proteins, and they come in different flavors and they come in different strains, a little bit about how are we going to characterize and then start classifying these different strains. So that when someone comes up and says, I'm looking at a PD alpha-synuclein folded strain, there may be 15 or 20 different strains at the end. You know, which one are you looking at, so that we can understand what the experiments are, but also better understand disease progression and rate, because these strains have different severity of disease and different rates of disease.
Yeah, and I agree completely. And it may be possible even from a therapeutic perspective that the way that you attack specific strains might be different. And so that could create a real challenge, and being able to really understand what you're working with will be really important.
And I think that's where biophysicists are going to be entering into our neurodegenerative space.
Yeah, and it's great to start, you know, with cryo-EM, start to really get some real good data on what these strains really are, you know, in addition to their behavior in vivo. And I think that there were some nice examples on both sides where John Collins, you know, really showed some data around, you know, and in using prion as an example, but then also moving on to A-beta, about really the specifics of what happens structurally, but then also with some of the scientists from University of Pennsylvania showing how these strains can behave very differently in vivo as well.
Yeah, that was, and then how they initially get folded. I was just thinking of Rudy Tanzi's talk where he showed that A-beta was a bacterial or pathogen binding protein, and that was quite scary, some of his.
Yes, and that'll be interesting to see. I mean, I think it's both an opportunity and a challenge to understand, you know, this growing literature that Rudy talked about around the interplay between viral infection and Alzheimer's disease, and whether that ends up being something that's correlative or potentially causative in disease.
Yeah, because we do know that it's genes and environment. You know, it's not just hand waving to say that.
Yeah, very true, very true.
It was also, I think, interesting, and I'm encouraging to see a number of these investigators are starting to talk about developing sporadic disease models. Yeah. And that's quite a challenge.
Yeah, and I think that's going to be a huge technical challenge. Very important, though, and I see from the therapeutic perspective, I mean, we're starting to develop good models based on genetic contributors to disease. But often those genetic contributors only contribute to a small subpopulation of patients. So understanding whether that biology applies to the rest of patients that present with these diseases will be extremely important. And I think it's not going to be easy to do.
No, but I think we have the technology to start doing those experiments and probing in that way. And it's great that we've got a group of really talented people who are thinking along those lines.
Yeah, definitely. I was impressed by, you know, a really robust group of young scientists at this meeting, and really seeing some of the next generation bring their ideas forward was really fun to see.
Yeah.
Also, I felt, though, from the therapeutic perspective, again, that's going to be super important for these, you know, innovation to continue to happen in the field, because, you know, although I think there's a lot of exciting new science going on, in terms of disease-modifying therapeutic approaches, they're really still lacking for a lot of the diseases that were discussed at this conference.
And it's clear from this conference as well that these diseases fall into subcategories. So even if you come up with a good therapeutic, you know, for Alzheimer's disease, it's going to be for a subset probably.
Yeah.
And so we're going to need to continue to funnel pipeline targets into therapeutics so that we can really get to meeting the needs.
Yeah, agreed. And there was some talks at this meeting. I'm thinking of the one from Matt Kennedy at Merck, who was talking about identifying the right patient population. And I think that's going to be a real challenge. I mean, in some cases, like he mentioned with LRRK2, you can identify patients with a LRRK2 mutation, but in other cases, it's not going to be that easy. And really, you know, developing better biomarkers to understand the patient population that you can match with that approach, I think is going to be extremely useful.
And also bringing in the clinicians who work with those patient groups, because there are very clear demarcations in early onset, mid onset, and late onset disease, but also in how fast patients convert. And so for a lot of the studies people will want to do, they're going to want rapid progressors and not slow progressors in a neurologic disease.
Completely agree, and it's going to be a big challenge. I think about Eric Kwong's talk that we mentioned earlier, and just in his introduction, thinking about progranulin deficiency and that only being a small proportion of frontotemporal dementia, but even within that disease, how many different types of pathologies and symptomatic onset there is, and there's really a wide amount of variability, and it's going to be very important to understand that on a molecular level so that we can really develop effective therapeutics.
And some of the speakers brought up using non-invasive imaging, but we're really trying to develop and think about developing ligands and imaging modalities to start to separate out those patients before they go to autopsy, where we can separate them out by other means.
Fantastic.
Yeah.
Well, I do have to say, this has been a very busy but a fun week.
Yes.
And I enjoyed, organizing this conference with you. It's been fun to hang out for the week and really hear some exciting science. And so I'm really looking forward to where the field is headed.
Oh, me too. And I had a great time. I couldn't have asked for a better co-chair.
Oh, well, thank you very much. I couldn't either.